Nick Lane, you have a very vivid picture in your head of where life first emerged on this planet. Paint that picture for us. Really set the scene.
Well, the kind of place that I think about are deep sea hydrothermal vents. And as soon as I say hydrothermal vents, most people probably, if they have a picture in their mind at all, it would be of some kind of smoke coming out of some chimney. These are the kind of things that we see on television. Um, that's not actually the kind of vents that I have in mind. The ones that we see today, and some people will be familiar with them, they are kind of tottering pinnacles of limestone, um, with all kinds of amazing kind of doodles of rock. I like to think of them as a Gothic cathedral almost. Um, it's a different kind of system. It's actually produced by interactions between rock and water, so it's not volcanic at all. And they're not obviously gushing out hydrothermal fluids into the oceans. Um, Now, inside them, there isn't a chimney, but you've got this labyrinth of interconnected pores. And the pores themselves, I mean, with a little bit of imagination, you can see them as cell shaped. They are kind of pockets with a barrier around them. And you have continuous flow going through. So there's two things that I would really take away from that, having kind of reflected on these things for decades now. There's two things. One is the structure. You've got a cell-like structure, and the other is flow, continuous flow past or over or through this cell-like structure. Um, not a not a really harsh flow, but just a continuous flow. So a continuous reactivity. And at the origin of life, you've got these reactive fluids which are being brought together with a structure which is facilitating their reaction and effectively converting gases into gases like hydrogen and carbon dioxide into organic molecules. And one thing which I've always found really gobsmacking, to be honest, is that if you've just got a flask of hydrogen and CO2 and you warm it up a little bit, shake it, and it's actually thermodynamically, in terms of the energy released and the entropy of the universe, uh, it is it is more stable and a higher entropy overall to make cells out of those two gases. Um, it will release energy into the universe if hydrogen reacts with CO2 to make cells.
It's
Um,
amazing.
that's just thermodynamically favored state. Then there's barriers. It's not going to happen very easily. But in these kind of events where the structure and the flow are breaking down those barriers, you're effectively turning gases into the molecules of life.
This is a different view than most people might have read in their textbooks. I remember back to my college lectures, and you hear about the primordial soup, right? The idea is like this beam of lightning, this initial spark, which might have just synthesized different chemicals, as you say, in the environment, and you get those original amino acids that eventually form RNA and all of the rest. How does your view differ, and why are you more confident in this hydrothermal vent idea?
Um, so it's a strange thing, but almost everybody's heard of the primordial soup, and
Yes.
most people are familiar with this idea of what was known as the Miller-Urey experiment, which was flashes of lightning, so electrical discharges in a flask, and that flask contained hydrogen and ammonia and methane and water and not a lot else, and the flashes of lightning forced them to react and form these amino acids, and this was back in 1953. This was really the first serious experiments done on the origin of life. And they had a very specific hypothesis behind it that the Earth's atmosphere would be something like Jupiter, um, which the gases were known roughly at the time. And so Stanley Miller had done these experiments and they worked astonishingly well. Um, Now, since then, there's been all kinds of squabbles about what was the Earth's atmosphere really like? Was it these Jovian gases or not? And the answer is probably not. Probably it was more like CO2. Um, the field has moved towards chemistry, for example. You start with cyanide and you use UV radiation, and then you've got a continuous reactivity there as well. The problem I have with all of this is you can make the building blocks of life, but then they kind of sit there, and the question is, and now what happens? And what animates it really?
Yes.
What makes them react with each other? And when they do react with each other, and the pathways required to make them looks nothing like biochemistry as I know it. And so you've partly answered the question. You made the building blocks of life, but they're inanimate almost. And the question is then, so how do they then come to life and invent all the metabolism and come up with a genetic code and all the rest of it? So they've not really answered the question. The other way of looking at it, which is my way of looking at it, as a bunch of people around the world see it this way, and it's always been disfavored by chemists because it's basically difficult chemistry. But start with CO2, start with hydrogen, start with the kind of catalysts that life uses. So iron sulfides, things like that, metal ions, things like nickel and molybdenum and so on. Start with those and potentially with some structure where you've got ion gradients across membranes, proton gradients. And what do you get? Well, the answer is you get the simplest kind of carbon-based molecules. Krebs cycle intermediates, slightly more technically, but basically these are the molecules that are just carbon, hydrogen, and oxygen. And from there, life makes amino acids, and we can do these things in the lab. And from the amino acids and sugars, you make the nucleotides, and we can do not all of it, but some of those things in the lab. So the idea, let's call it imagination still, but this is the aspiration of what we will be able to do in the end, is you start with CO2 and hydrogen. And before you've got any genes or any genetic information or really any proteins or enzymes or catalysts of that sort, you get a kind of network of reactivity which foreshadows metabolism as we know it. And so all the stuff that's happening in your cells right now was happening back then in a hydrothermal system, just because it's thermodynamically favored chemistry. That's what happened when you get these molecules to react. So we're still a long way from doing that, but over the last 10 years or so, the evidence that it's not completely unreasonable has got a lot stronger. I think a lot of people would have reasonable doubts still, but certainly it's much stronger rather than weaker. And once you've got there, then the first genes, all they actually need to do is amplify flux through this pre-existing metabolism. Whereas if what you have is information first, well, what do the genes have to do? They then have to encode all the metabolism, and that's really tough, actually. That's not something that anyone's ever got near to doing.
Right. You have a great quote in the book, which I think sort of echoes what you said in your book, Transformer. The fit between the geochemistry of alkaline vents and the biochemistry of methanogens and acetogens is so close that the word analogous does not do it justice. It's really remarkable that you're saying that the earliest system was 100% geochemistry, was environmental components interacting with each other. And then somehow we got this local biochemistry. How do you get that local chemistry not to dissipate to zero? How does entropy not just do its job and just squash all chemistry that emerges? How long can that chemistry exist locally without just being crushed? Do you know what I mean?
Yeah, I mean, the answer has to be indefinitely. So the question is that real? So I'm talking about these cell-like compartments, this labyrinth of interconnected micropores and so on. If you think in terms of the thermodynamics, I said it's favored to get whole cells, but there are certain molecules that are more favored than others. The ones that are most favored are amino acids and fatty acids. So these are the ones that if you're reacting CO2 and hydrogen, you should get most of those things. You should get fatty acids and amino acids. And things like nucleotides to make DNA or RNA are a bit more difficult and complicated to make. So fatty acids, we and others have shown that if you put them under those kind of conditions, they will spontaneously form a bilayer membrane, very similar to the membranes you see around cells today. So they will form what I'd like to call a protocell. It's basically a kind of a membrane around an aqueous space. And they are pretty stable things. How long will they last without dissipation? It's difficult to know. But what we're really looking for is not a system which is never going to dissipate so much as a system which is continually being replenished. So you have dissipation continuously. You're going to be losing things. They're going to be reacting, moving on, being flushed out of the system. But you're also because of this continuous flow, you're continually making more of them. And the ones you make more of are the ones which are the most favored. And so you're effectively, you're in a steady state that you're continually making these things and continually losing them. And on balance, the kind of steady state equilibrium is you've got cell-like things, and those cell-like things are continually potentially converting more of these gases into more interesting things inside them. So as soon as you've got a cell-like thing, then you're beginning to prevent the dissipation already.
Right.
The membranes themselves will tend to stick to the mineral surfaces. So they're not going to just get flushed straight out into the ocean. They physically just sit there inside. I imagine them almost templated within the pore as a kind of a pore-shaped vesicle, proton cell sitting inside it.
Is that possibly a good argument for why you think that life emerging is so much more probable in the hydrothermal vent system? Because if you have a primordial soup, it seems a lot less likely that it would ever persist consistently and ever be able to persist at a greater rate than it would just be dissipated if you don't have these geochemical structures which are creating protocells and membranes and local chemistry, that it would be much more likely it would dissipate to zero within this soup. It's just too chaotic, it's too messy.
I mean, even within a soup, you're still going to have a formation of new materials and a kind of elimination of older materials. So you'll still have some turnover, but it's not very obvious where the structure comes about,
Yes.
or why something would grow.
Yes.
So if you think about, what cells do now is they grow, they double. One cell makes 2 cells, makes 4 cells, and so on. And inside those cells, you've got the genes, and the genes are made of DNA, and the DNA is being copied, and each daughter cell gets a copy of it, and so the DNA also is going 1 to 2 to 4 to 8, and so on. Now, that's a form of doubling as well. It's just more exact than a cell kind of just falling in half, you might say. And I mentioned these fatty acids, membranes, protocells. They're very dynamic things. They do fuse together and divide in half and so on. So there's no problem with them making copies of themselves, very approximate copies, so long as they're able to grow. So why would they grow? Well, they grow because you're continually replenishing the reactive requirements from the environment. And so you're continually making more stuff. And that stuff is apportioning itself and dividing and fusing. And on average, you end up with making more and more all the time. So growth is really coming before the more exact forms of growth, which would be replication of DNA. But
Right.
First of all, you need to be making more stuff all the time. Now, that's what I don't see happening very easily in a kind of any soup-like system. What is bringing it together and making these things make copies of themselves? I just don't really see the driving force there. Whereas in a hydrothermal vent, you can't escape the driving force. You're constantly converting this flow of stuff into more of yourself.
That makes complete sense to me. Explain the alternate hypothesis of this RNA world. If metabolism wasn't first to emerge, the original idea was that RNA emerged, and that was both the information scaffolding as well as the catalytic engine, that it could replicate itself. Firstly, I don't really understand how it's able to replicate itself with any level of fidelity. It seems like you just make a lot of mistakes and then it might go to zero. But explain that a little bit. Explain that hypothesis first and why you think it's less likely.
So the hypothesis is beautiful. And it comes about really from a chicken and egg problem with DNA, because
Right.
DNA is the genetic code, but it's basically inert. And what it does is it codes for proteins, and proteins do all the work, but they're coded for by DNA, and they can't make copies of themselves. And so there's this question, which came first, the DNA or the proteins? And actually there is an intermediary, which is RNA. So DNA makes a kind of carbon copy of itself almost, which is called RNA, which is a short form of it. And that codes for the protein. Well, RNA also can fold into interesting shapes and has catalytic potential. And so it can do partly what a protein does and partly what DNA does. And so there was this idea that before you had proteins and DNA, you had a world in which RNA was copying itself and doing chemistry. And it's a simple idea.
Yeah.
It's never been demonstrated to be true, but there's been lots of people working on, can we make a ribozyme, so an enzyme which is made of RNA, that's capable of copying itself. And to some extent it's been done. It tends to be this one will copy a different one, which copies a different one, and you can go in what's called a hypercycle and eventually things will copy themselves. But it leads to downstream problems, and in terms of how do we get to where we know the end point is from there. So what would an RNA world have to do? Well, it would have to invent cells. It would have to invent metabolism. It would have to invent proteins and the genetic code whereby DNA codes for proteins in the end. So these are the things that the RNA world has to do now. And actually, almost none of that has been demonstrated in the lab as being real. It's actually almost being completely ignored. So the challenge for an RNA world really is to get from there to life as we know it. And there's kind of an assumption, and it's a very lazy assumption, that if you've got genes, if you've got RNA which can copy itself, you've got genes. And if you've got genes, well, then natural selection can happen. And once you've got natural selection, you're away and you don't need to explain the rest. It's just a detail. It's really not a detail.
Right.
It's, there's selection can work in multiple different ways, but there's also things it can't do. And there are famous things that it can't do. And one of the more famous ones is come up with complexity, because the point about little bits of RNA, little genes which are copying themselves, is they will tend to get smaller and faster. They're basically selfish. They will copy themselves furiously at the expense of any kind of cooperation or altruism, you might say. Cells put them together in the same bag. So now all of these selfish bits of RNA, well, they can't get out of the cell. They've got to cooperate in some way and help the cell to grow. But now we're in a very different context.
Right.
So the end points depend on really what the starting point was. And if the starting point is you're already in cells and you can already generate the materials needed to make a gene, then you're all in the same boat, and selection is going to operate very differently to if you've got what people think of as a naked RNA world. Naked meaning lots of little genes all kind of squabbling and fighting between themselves, but not inside cells.
Right. Well explained. I just love the way you do scientific communication. I think it's so well done. Okay, so what we have is we have carbon dioxide and we have hydrogen, and they're fusing together with the work from free energy produced at hydrothermal vents into organic matter. Is that right so far?
Basically, yes. Yeah.
Okay. And then that evolution of that system, is that going to drive the evolution of the information, the RNA and the DNA? Is that metabolism going to drive that evolution? Are they co-evolving in some way?
Yes, I mean, this is some of the most beautiful work that I've been involved in over the last few years. These ideas were really coming from Stuart Harrison in my group, and for decades actually we've known that there are patterns in the genetic code, and these patterns suggest some link with metabolism and some link between the genetic code. You've got what are called codons and anticodons. So the codon is a triplet of 3 letters in the genetic code, which codes for one amino acid in a protein. So if you've got a stretch of DNA or RNA, you think of it in groups of 3. You can think of it as a long ticker tape of letters grouped into units of 3, and each unit of 3 is called a codon, and that codes for one amino acid. If you zip up all the amino acids, then you get a protein, and the protein, the sequence of amino acids in the protein depends on the sequence of letters in the DNA grouped into these brackets of 3, which is the codon. So that's the genetic code in a nutshell. And the thing which is odd, there's various patterns, and one of those patterns, for example, is that the first letter of this codon corresponds to the position in metabolism, more or less, especially if you look at it as, let's say, metabolism starts with CO2 and hydrogen, and you go through metabolism as we know it. How long does it take you to get to this amino acid or that amino acid? Some of them are closer to the starting point of metabolism, some are further away. Well, that corresponds to the first letter of these codons. You can plot a graph, and the ones which are close to the beginning of metabolism, they all start with a G, and the ones which are furthest away, they've all got a U at that first position. So it's kind of odd. What is it? How do we interpret that? But it's there for sure. The second position corresponds to the physical properties of the amino acid that's coded for. So especially, is it hydrophobic or hydrophilic? Is it water loving or water hating? And the more hydrophobic letter that's there, base that's there, so the least water loving one corresponds to the more hydrophobic amino acid. So the two things sit together. They're physically comfortable together. And correspondingly, the other way around, the more hydrophilic one, so uracil is the most hydrophilic base, goes with the most hydrophilic amino acids. So it just looks like they're nestling up together, snuggling up together physically. And then the third position is about the size, just how big are these things that are nestling and snuggling up with each other. So all of that says this, in the broadest brushstroke, the genetic code has got patterns in it that suggest there's a link with metabolism, and these things were interacting directly for physical reasons right at the beginning. And what that suggests in turn is that metabolism came before the genetic code, that these things were co-evolving, really, as you put it. Exactly how? I mean, we've been trying to do models on this kind of thing to show that you can introduce a random sequence of RNA, and over multiple generations, it will eventually end up coding for an optimal peptide, optimal protein. Is it real? Is it true? All you can do with a model is just show that it's systematic, really, that this thing is possible under these conditions.
Right.
So it's hard to know, but what the really exciting thing about it is that information, the origin of genetic information, has been such a problem in biology that brilliant minds like Paul Davies and Sarah Walker and people like that are trying to call on some unknown law of physics to explain where information is coming from, whereas in fact all you really need is physical interactions that say you've got a random stretch of RNA and it's going to template through physical interactions a non-random sequence of amino acids in a peptide. If you zip them all up to make that peptide, then the sequence of the peptide depends on the sequence of the amino acids. Sorry, the sequence of the letters in the code underneath. And so then you have selection as we know it in a cellular context with a metabolism as we know it. And in this case, you really do have selection for greater complexity. So it may not be true, but it has huge explanatory potential, and it's consistent with everything that we know about the genetic code.
Do you think we'll ever conclusively prove it? Is there an experiment that can be done in the lab or on a computer with some advanced AI and computational models, and then we'll be like, bam, that's it. We have solved it conclusively.
No, I don't think we ever can do that.
Right.
In principle, we could. It certainly wouldn't be as simple as ever simply proving that this is how life started, because that's a kind of a historical question. The only way it could be proved that this is how life started would be to disprove every other hypothesis as to how life could have started, to say, no, at this point it gets stuck, no, at that point it gets stuck, and so on. So I don't think we're anywhere close to doing that. If there is only one path to go from prebiotic chemistry of a specific kind to cells as we know them, and everything else cannot possibly work, then it would stand to reason that life must have followed that path.
Right.
But I think we're a long, long way away from doing that. But I think that it's a long path, and maybe there's multiple ways through it. But if you start at the beginning and you specify an environment, then you're obliged all the way along to say, okay, well, if this is true, then here's what I should get, and if that's true, then here's what I should get, and the end point has to be where we know it. And if you can't get there, then your ideas are obviously wrong.
Right.
So I was criticizing the RNA world. Can this RNA world invent metabolism? I think the answer is no, but I'm not going to try and prove that, because that's their job. People who believe that it can, that's their job to prove that it can.
Has anyone came up with an alternate algorithm for life, like a system that seems to work from the start based on the environmental components that does exist? Or did it have to be the way it is?
I think that depends a great deal on what you would mean by life. So if you were to talk about, say, computational forms of life in silico, then it's probably been done already. There will be things that are lifelike and can evolve, and so in that sense, it's trivial and it's being done. if we're talking about organic life or life on a planet, so it's not actually brought into existence by someone inventing computers and all the circuitry and everything that goes with it. If you start with a planet or a moon or space or a sun or wherever it is, any environment in the universe, how does life bootstrap itself up from effectively inorganic chemistry to whatever life might be? I think there are serious constraints on what can be done and can't be done. And I'm sometimes accused, and fair enough, of being kind of narrow-minded. And the criticism that will come is it's an infinite universe, an infinite number of things can happen, and you've got a little brain, so and you can't possibly imagine all of the things that could be happening out there. So stop being so arrogant and dismissing, the possible amazing nature that life could have. It may be true.
Harsh criticism.
Well, I don't think it's a scientific criticism, because science is really about, can you test things? And it's no good just saying, oh, well, it may be amazing. And that's not really science.
Yeah.
The question is, we only know of one form of life. And all life on Earth is basically the same insofar as it's carbon-based. It's cellular. And some people will immediately say, but viruses aren't cellular, but viruses depend on cells to copy themselves. So in that sense, we can reduce it back down to life is cellular. It's carbon-based, it works in water. And so then the question is, did it have to be that way, or could it have been some other way? And the more I've thought about these things, the more I think there's very good, strong reasons why it is the way that it is. Carbon is extremely good at this kind of difficult chemistry, and it's extremely common. It's one of the more abundant elements in the universe, and it's far better at that chemistry than, say, silicon is, and it's far more abundant than silicon is. And on Earth, silicon is much more abundant, but all life is carbon-based. It kind of says, forget about silicon, it's not the answer. For water as well, it's an astonishing solvent with properties that we barely understand even now. The way that it interacts with proteins, the way that it interacts with membranes, with transfer of protons. It does all kinds of things that most other solvents just don't do. And again, it's abundant. Hydrogen and oxygen are abundant, and water is abundant. So just on a statistical basis, we're likely to be dealing with the same chemistry anywhere else in the universe. And then the question is, under what range of conditions will you be able to react CO2, which is again the dominant form of carbon, into organic molecules? And that range is not huge. You're dealing with, between, more or less the range of the freezing to boiling point of water, and not much more. Under those conditions, you will get organic molecules. So it may be limited, but I think in some sense the onus is on the people who think that life could be any way you want it to be, to say, okay, well, here's how.
Yes. Yes.
And I think we can try to look at life on Earth and understand why it's this way. I mean, the subtitle of the vital question was, why is life the way it is? And it's just a really interesting question. Did it have to be this way, or could it have been a completely different way? And the more I've thought about it, the more I've thought there's really good reasons why it's this way.
Yeah. If you knew for sure that there was carbon dioxide, hydrogen, water, and some sort of proton gradient on a different planet, would you then say with pretty high certainty that you think there's life on that planet?
I would be optimistic. I would add another couple of things in. I mean, I'm talking about continuous flow hydrothermal type systems. So what does that require? That requires what I'd like to think of maybe a bit romantically as a living planet, which is to say a geologically active planet, which is to say the crust is turning over continuously. And what that's doing, so on Earth, basically all the iron has gone to the core and to the mantle. And so the Earth is a giant battery. Iron is very electron rich. The atmosphere and the hydrosphere, so the oceans and so on, are relatively electron poor. Volcanoes are outgassing stuff, but the temperature of volcanoes means what they're putting out is mostly carbon dioxide and water vapor and things like that, not hydrogen and ammonia. So it's water and CO2 that's coming out of volcanoes mostly. So relatively oxidized because of the temperature and because of the sun. So you've got a battery. The outside is electron poor, the inside is electron rich, and the places that connect them are the hydrothermal vents, and that's where all the action is happening. And if that reactivity begins to cease, and it would cease if you're not turning over the crust, because you know if you don't have plate tectonics of some sort, then the processes I'm talking about, effectively the water is reacting with the rocks. The technical term is serpentinization, but it's basically rock-water interactions. That reaction would go 5, 6, 10 kilometers deep in about 1 million years across the entire surface of the Earth, and then it would stop. And so to get it going again, you need to be subducting some of this crust down into the mantle or even lower, and bringing up fresh mantle rocks to the surface. So you've got what amounts to a living planet. It doesn't have to be modern plate tectonics as we know it, but you've got this continuous turnover. And it's really, that system is driven by gravity as much as anything else. It's not really driven by the sun. It's driven by the fact that all the iron went down into the core, and so you've got a battery structure.
And how specific do you think that ingredient list is? I spoke to Dr. William Radcliffe on the podcast, who studies the origin of life and complexity. And he kind of dropped a line, which I thought was pretty shocking, which was he doesn't think life is that unlikely. He was kind of shrugging his shoulders like, yeah, it will emerge when the things are there. Like, how specific is that ingredient list, that active planet that you just laid out?
I would say there would be tens of billions of planets with those conditions, planets and moons, in the Milky Way alone. So common, I would say, among the more common types of planets. But I don't really know. I'm not really an astrobiologist, but I think those kind of conditions would be pretty common. Now, how likely it is that you go all the way to getting cells with molecular machines, with DNA, with, it's very hard to know, but my hunch is it's all favored on a planetary scale is going to happen. And actually my hunch, which I find a little bit scary, is that you would get cells that resemble bacteria, and they're going to be of a similar size. They're going to be carbon-based. They're going to have similar biochemistry. They're going to have similar patterns in the genetic code, not necessarily the same genetic code, but the reasons it's templated here through direct physical reactions. If what you have is a set of conditions that generates metabolism spontaneously all the way up to nucleotides, then you will get the same metabolism and the same nucleotides under similar conditions starting from CO2 elsewhere, and then you'd end up with a genetic code that resembles the one that we have. And I find that quite a scary prediction.
Right.
It doesn't feel a comfortable place to be, but it appears to be the only reasonable prediction for the arguments that I have been making. And I think the arguments are quite strong.
What's most scary about that?
It's the level of determinism. It's a little bit reminiscent of the cosmological constants. That if the strength of the gravitational force or the strong force or the weak force or whatever it is was slightly different, then you would not have a universe with suns in it, with gravity as we know it, with the lumpiness that's required, to form stars, to form the elements, to form a universe capable of giving rise to life. Change those cosmological constants slightly, and you end up with something that never forms stars or whatever it may be. So the physicists have dealt with this by effectively conjuring up a multiverse, where they say, one way of doing it is to say, they happen to be just right in this universe. They would have to be because we're here. But in the next door universe, they could be very different, and it could be a very different place. And the idea of the multiverse is carried from quantum mechanics. It's a fascinating idea. You can make very, very entertaining movies about it. And it seems to solve that problem of the cosmological constants. It's either that, or there's a fundamental reason for why they are the way they are. Now, let me bring that back into biochemistry. What I'm saying is you start with CO2 and hydrogen and water, and you're going to get cells with a genetic code that resembles ours. it feels fine-tuned to the kind of chemistry that's going to inevitably give rise to the same kind of life again and again and again. So it feels as if the universe has been fine-tuned to give rise to life at the level of biochemistry as well as at the level of the cosmological constants. But here, there's no way that I can call on a multiverse because I'm dealing with the laws of chemistry as we know them. I can't explain the propensity of CO2 to give rise to metabolism and genes in the way that it seems to do by saying, oh, it happens to be this way in this universe. No, these are the laws of chemistry that make it happen that way. So why are the laws of chemistry that way? At this point, I'll kind of shrug my shoulders and hand over to the physicists. But
Right.
I do find that level of determinism. You may just as well say God did it.
Yeah.
I would say, and I would. I'm not a religious person, but I would tend to use the word God and thermodynamics almost interchangeably to reflect forces that are acting on how substances behave in ways that I don't really have a clear understanding of how those forces came to be.